प्रथमेश.
हा एकच मिनिट झाली सर.
Good evening, sir.
Good evening. Good evening.
Sir, how's you?
Yes, I'm very fine.
Okay, thank you, sir. So thank you for providing me this opportunity again.
Yes, no, you are doing good thing.
Yeah,
So I
thank you, sir.
must provide the opportunity under the guidance of Pine. You are doing the best thing.
Yes, sir. He's a very active person, and I am in touch with him since last 1 year. So he's doing
Yes,
well,
yes.
quite well.
Very.
Yes, sir.
Yeah. So you can start now. Whether a speaker has joined.
Sir, just give me a minute. One of my anchor is unable to join. Yeah, I will add her. Yes. So she is now in. Miss Debarpita, are you able to hear me?
Hello.
Hello. Yes. Miss Debarpita, please switch on your camera and please start the meeting.
Yeah, sure. Has everybody joined?
Please start the meeting, Miss Debarpita.
Okay. Am I audible?
Yes, ma'am.
Yes, होता है. Normally, post office PO होता है. Good evening, everyone. Today, we gathered here for the international webinar on the topic Agricultural Biotechnology that is organized by the Microbiologist Society of India. Today, we have with us our esteemed keynote speaker, Dr. Khushbu Jain, Assistant Professor of Department of Biotechnology, Faculty of Science of Janardhan Rai Nagar, Rajasthan Vidyapeeth, Udaipur, Rajasthan, who has accepted our kind invitation to share her knowledge and expertise. We are highly thankful to the organizing board members. Firstly, the Chief Convener, Sarat Subra Pai, the National Representative of the Microbiologist Society of India, Professor Dr. Arvind Deshmukh, Professor of Emeritus and the former Head of Department of Microbiology, Dr. Baba Saheb Ambedkar, Marathwada University, Maharashtra, the founder and of All India Chairman of Microbiologist Society India, who has helped us in arranging this international webinar on agricultural biotechnology. Dear all, we all know that any event is not going to be successful without praying to God. Therefore, I, Debarpita Das, a student of the Department of Biotechnology at St. Xavier's College, Autonomous, Kolkata, your host for today, want to do a prayer service. Let us bow our head and put ourselves in the holiness of God, the Father, the Son, and the Holy Spirit. Dear Almighty and ever-loving God, we glorify and thank your name. You have showered us with so much blessings, and your presence continuously reminds us of your faithfulness and guidance. May we humbly ask you to shower our speaker today of your greatest inspiration so that he may share the most of his knowledge, his field of expertise, and to his respective topics. May we also absorb the invaluable knowledge, expertise, and experiences and put it into practice what we may learn today. Your infinite blessing would mean the success of the seminar. May we be a living witness. Hello.
Hello. Miss Debarpita,
Yes.
is it over?
Yes, your infinite blessing would mean the success of this seminar. and may we be a living witness of your guidance, love through the enactment of the knowledge acquired through this activity. We ask this in the name of Jesus Christ, our Lord and Savior.
Yes. Now, please, Miss Sumbul, are you there? Miss Sumbul, are you there? Uh, I guess she's not here. Okay. Miss Devarpita, please welcome first Dr. Deshmukh, sir. You have the thing. Miss Devarpita, hello.
Yes, sir. Just a minute. Welcome, sir. Professor Dr. Arvind M. Deshmukh, Professor Emeritus and the former head, Department of Microbiology of Dr. Baba Saheb Ambedkar, Marathwada University of Maharashtra and Government of Maharashtra. He's a founder and All India Chairman of Microbiology Society, India. Welcome, sir.
Thank you, Devaprita. Can you hear me?
Yes, sir.
Hello. So you can hear me. Okay, thank you. Thank you. In fact, you are arranging such lectures regularly. Now you are habitual to arrange such nice lecture series. The young generation of India, I can see in you, 100 students are attending and many more will be attending on YouTube. So that's a really good thing for the microbiology and for the biotechnology. And today's speaker will be talking on a very, very important topic, agricultural biotechnology. So the madam is from Udaipur, Dr. Khushbu ma'am. And really she will be focusing light on the agricultural biotechnology. As a president of Microbiologist Society, I will only appreciate all team worker, all students. They are regularly contacting me. And Paine, who is the convener, main convener, under the blessing of Dr. A.K. Srivastav, sir. So I wish again all the best to all of you. Thank you. Thank you.
Thank you, sir.
Yes.
Thank you.
Now, introduce Dr. Khushbu Jain, Miss Rashi. Miss Rashi, are you there? Miss Khanna, are you there? Are you able to hear me? Miss Rashi,
Yes, sir.
Rashi Khanna,
Good
are you
evening,
there,
sir.
Miss
I can
Rashi
hear you.
Khanna?
Can you hear me?
Yes, go ahead.
Sure, sir. Good evening, everyone. So my name is Rashi Khanna, and I am one of the hosts for the evening for this event. I am currently pursuing BSc Biotechnology honors from Punjab University, and I would love to give an insight on Dr. Khushbu Jain, who is our key speaker for this event. Dr. Khushbu Jain is an assistant professor in the Department of Biotechnology, Faculty of Science at Janardhan Rai Nagar Rajasthan Vidyapeeth, deemed to be University Udaipur. She holds a BTech in Biotechnology from Sir Padampat uh, Singhania University, Udaipur, and MTech in Biotechnology from DY Patel University, Navi Mumbai, and a PhD in Biotechnology from Mohanlal Sukhadia University, Udaipur. With a strong academic background and extensive research expertise, her primary areas of interest include plant biotechnology, microbiology, bioinformatics, and the applications of machine learning and artificial intelligence in the field of biotechnology. Dr. Jain has contributed significantly to academia and research, having published 13 research articles, 30 book chapters, and 2 books. She has actively participated in national and international conferences, presenting her research and receiving awards at 13 different events. Additionally, she has served as a visiting faculty member at Amity University, Mumbai, Shri Padampat Singhania University, Udaipur, and Mohanlal Sukhadia University, Udaipur, gaining diverse teaching experience. Beyond her academic and research contributions, Dr. Jain is also involved in the editorial and review services for Iterative International Publisher India. Her excellence in research has been recognized with prestigious awards, including the Young Researcher Award 2022 by the Institute of Scholars and recognition among the top 100 in the Augmentin Writing Skills for Articulating Research competition organized by the Department of Science and Technology, Government of India. Through her interdisciplinary research and teaching, Dr. Jain continues to contribute to the advancement of biotechnology, fostering innovation and knowledge dissemination in her field. With this, I welcome Dr. Khushbu Jain, and over to you, ma'am. Thank you so much.
Thank you. Thank you, Rashi. So I'm very thankful to the MBSI team, Sir Deshmukh sir, Sarat, and his organizing team members to invite me here for the presentation on an international webinar on agricultural biotechnology. So most of the participants here I am seeing most of the participants are from the life science background or the biotechnology and microbiology students. So it is very nice to talk to them that what is the role of the biotechnology or agricultural biotechnology. Here I am starting with my presentation. Sarat, please let me allow to share the screen.
Okay, I will. Please check.
लाइव साइलेंट होना क्या म्यूट कर लो
Right, Khushbu ma'am, are you there? Please switch on your microphone.
Yeah.
As of now,
So
first of all, I will mute other participants. So that time your mic will also be muted right now. Again, I will give you an option to unmute, then you will proceed.
okay.
So you see, right now I'm muting everyone. Yes. So right now everyone is muted and they cannot unmute themselves even if they want. So I'm giving you an option to unmute yourself. Now you
Okay.
just click.
Yeah, I have unmuted
Yes.
myself.
Now only you will be speaking. You will be visible. You are co-host now, and you will be able to share your screen, which is visible to us and visible to.
Okay. So my screen is visible to all of you.
Yes, yes, it is visible to everyone, so you can proceed.
Yeah, okay.
It is visible.
So here I'm going to start on the topic of this on the advancing agricultural biotechnology through agri-informatics. So here are the two terminologies are residing in this title. First one is the agricultural biotechnology and second one is the agri-informatics. Agriculture, when we talk about the agricultural biotechnology, the first thing which comes in our mind, it's related to the plant tissue culture or related to the gene editing, gene silencing, and the micropropagation, and all these things comes in our mind. But beyond that, nowadays, what is the scenario is going on that the continuously climate change is going on, and the continuously demand of the food is increasing as the population is increasing. So there is the need to enhance that enhanced technology. Although the agricultural biotechnology is the enhanced version of the basic traditional farming, but there is also an enhancement in this technique that is known as the agri-informatics. So I'm going to focus on this in this part of this title, that is the agri-informatics. So if we look into the background of the this, then can I minimize this? Yeah. If we if we look into the background of this topic, agricultural biotechnology, so we need to understand why it has been introduced earlier. Because the if we look into the statistics of the which has been published by the FAO, Food and Agricultural Organization, or the data collected from the Indian Horticulture Database and the other agricultural societies, they promises and it ensures us that the 40% of the country's populations which are directly or indirectly involved in the agricultural background. India is the one of the most largest producer of the food in the world, and it produces the 329.7 million metric tons in 2022-2023. And this data has been gathered from the Food and Agricultural Organization. This demand is the as the population is increasing and there is the sudden increase in the population globally, the demand of the food and agricultural is increased and will be increased by the 70% by the 20 to 25 to provide the 9.6 billion people to feed the 9.6 billion people. So there is the huge requirement or tremendous requirement of the food production that everyone should get feed. There is the definition of the feeding, not eating the food. It will get the nutrients as well. Nutritive foods should be provided and should be collected by the people to get live a happy life. So there is the need of the so scientist has developed the advanced technology of biology, that is the biotechnology, and that another stream has been introduced, that is the agricultural biotechnology, that is known as the which enhances the crop yield, pest resistant, livestock, and sustainability. Ultimately, the main aim of the to introduce this such type of field is the increase in the crop yield. To address this, what are the different parameters which are the and problems which has been addressed by this field? That is, first one is the food security. As I said, everyone should get not only the food but it should get the nutritive food. That is climate resilience and sustainable farming. Sustainable farming means that whatever we are producing, each and every part of that plant should be utilized and farmers should get most output from that farming. The techniques used in the agricultural biotechnology since past area, that one is the gene editing, gene transfer technology by the agrobacterium transformation and micropropagation, somaclolan variation, synthetic seed propagation. As you, I think most of the students and faculty members are involved are from the biotechnology and microbiology background, so they all know about these techniques. Uh, since the past era and since now till now we are using these techniques to over for the various research purposes to enhances one of the example of the gene transfer technology of by the agrobacterium transformation is the bt cotton and golden rice, which is enriched by the vitamin A. So these are the some techniques which are used through the micro propagation. We were able to propagate those varieties which are seedless varieties like banana. So through this technique, we are enhancing our crop yield. But nowadays, then the recent techniques, but still there is the demand to develop these techniques also. So the recent advanced techniques which are used in the field of the agricultural biotechnology is one is the gene editing, then the next generation sequencing, synthetic biology, biologicals, and the bee vectoring. Here we are using the some of the techniques like the CRISPR-Cas9, the method. This is a technique which is used by most of the scientists nowadays, that they are not transferring the useful gene or the whatever the gene is required, active gene in the another species. What we did in the what we do in the agrobacterium transformation. Okay, what we do, we take a active gene and transfer it into the in the our vector, and through the vector we transfer it into the our plants, where it enhances its productivity when it gets activate. It specific genes get activate that produce the active protein from that. But in this case, CRISPR-Cas9 method, or if we look about the RNA interference technique, then in that case, we are silencing the genes. We are those genes which are responsible to enhance the stress in the plant, which are responsible to attract the insects and to get a disease in themselves and to get diseased in the plants. Those genes are being silenced and the new variety has been developed. So one of the GMO, genetically modified free plant, when we introduce a specific and through the technique agrobacterium, this the after the output product, the output yield is known as the genetically modified organisms. But here, using these techniques, these are exogenously applied as the GMO free, that is not the genetically modified organisms. We are not introducing any genes, but only we are modifying the genes which are present in the plant itself. Then the biosensor techniques. Biosensor is the most prominent field in the world, in the field of the in the agricultural side, that most of the scientists which are using the electrochemical transmitter sensors, which to gather the real-time data, what is going on in the plant inside or in the vascular tissue of the tree. So what they are implementing electrochemical transistors in the plant itself and producing, and they are getting the data from that how much sugar is produced by the plant if it is under the stress condition, how much if it is the fruit is in the ripening condition, they produce the ethylene more. And the many times when the plant under the stress condition. Stress means there are the two types of stresses, biotic stresses and the abiotic stresses. If the insect get attacks on the plant or the plant any other crop variety, then the plant produces some type of regulators or some type of metabolites to to fight against that insect or that parasite. So how that level can be checked by the use of the biosensors. Then another technique is used in the recently nowadays they are using the 3D bioprinting. If we talk about the agricultural biotechnology, we should not forget about the terminology of the crop yield, enhancement of the yield in the livestock also. It comes under the agricultural part itself. So there is the 3D, by the use of the 3D bioprinting, we can use the ribeye stick, wagyu beef, chicken pieces has been grown, has been cultured artificially at in vitro level, and the ribeye stick chicken pieces has been developed. But there is a large controversial and ethical economical problems, the public health concerns are associated with this type of technique, as it the whatever the component has been, that the controversy is related nowadays like the same that what has introduced previously with the 3D with the pt brinjal or the bt cotton. Then the biologicals in another word we can say these are the chemical protectants, or the you can say the biopesticides, bioinsecticides. These are the materials which are produced by the um, um, biological molecules when or biological miniatures like the bacteria, so many bacteria, fungus, and the viruses are there which are able to produce the plant protecting chemicals. This biological that like the bacillus species, pseudomonas species, and so many species have produced the specific type of phenolics compounds and the alkaloid compounds which have the antimicrobial activity, anti-insecticidal activity. So this type of molecules which are derived from the microorganisms and the plant itself, then this comes under the biologicals. So these are the like here one example has been set for the insect pheromones. Like the plant produces a specific type when the rhizobium is attached in the roots of the plants. There is a specific type of pheromones has been generated and released by the root itself. They get attract the insects. The insect get consumed the specific type of micronutrient, whether it is the calcium, magnesium, and provide the availability of the other nutrients, and so the plants are able to accumulate other type of, not the single type of nutrient from the soil, but can absorb the other type of nutrients from the soils also. So, insect pheromone is the one of the naturally occurring non-toxic, non-persistent molecule that can serve as the crop protectant. But the limiting factor of this insect pheromone is they cannot inhibit the another insect. They can attract only one type of a specific type of species of the insect. They cannot target the large quantity of the insects together. So this technique has the limiting factor here, but the in the case of the biologicals, like we are, if we produce about the microorganisms, whatever the alkaloids and phenolics and the volatile organic compounds which are produced by the microorganisms, it can be a have the good range of the comes under the biologicals. Here is the diagrammatic representation of the 3D printed cultured food. How this ribeye chickpeas has been introduced when that it got the muscle cells, isolated the somatic cells or the mixed cells, muscle cell types. In case of the somatic cells, we go with the IPCS transformation. IPCS is the induced pluripotent stem cells, where the stem we are using the somatic cells and it is converting into the stem cells, and stem cells has the ability, has the pluripotent tendency, so they can get convert into the any type of cell, whether if it is whether they can convert themselves into the liver cells, heart cells, muscle cells, any type of the cells they can convert themselves. But in case of the so this is the stem cell biology. Through that we are differentiate after the differentiation and culture technique, we are getting this type of mass of the cells. But in case of this by using the 3D bioprinting material, we are using mixed type of cells. So we are using the 3D bioprinter, we are culturing and getting this type of muscle cells. After the differentiation, here initially we are getting the mixed type of culture, so we need not to transfer the cell into a specific type of the multipotent in a specific type of direction. Okay, so here is the limiting factor of this one, how we can get the multiple differentiation in this somatic stem cell technology. The challenges faced by the recent technology, although these are the recent technologies, but there are the challenges associated with it. First is the data overload, that the how throughput sequencing, sensor data, remote sensing the images are huge, and we require a specific resolution in that. Only that after that only we can get a specific solution. If we do the data analysis and sense imaging imaging technique is there to recognize the whether the some of the farmers are using and some of the scientists are using the imaging technology to identify the which of the disease is there in the plant. Okay, so we require many a times it happens that two diseases has the similar kind of the initial stage. They have the similar kind of that you can say there is a similar kind of phenomenon has been represented by the two different types of microorganisms. So we can't say that whether it is a bacterial disease or it's a fungal disease. After the when a pathogen attacks into the I want to say, let me describe in Hindi that if अगर एक पैथोजन किसी प्लांट को अटैक करता है, that time पैथोजन की जो भी उसकी जो भी उसका क्राइटेरिया है, that is there is the life cycle goes inside the plant itself. Okay, so initial stages जो भी रहती हैं उसके अंदर हो सकता है कि इनिशियल जो भी उसके जो भी इमेजेस हैं या इनिशियल जो भी उसका मेथड है वो सेम हो, whether it is the bacterial disease or the fungal disease. So in the data overload condition, the remote sensing images, we need to provide a specific type of image to clear identification of the disease. Then the climate change, it has happened, is the globally affecting the crop yield. There is the stresses are going on, there is the lack of the like the nutrients are available in the plant materials and the soil. So there is it's a big challenge to the farmers. Then the consumer acceptance and regulations. Consumer acceptance is a big demand. Although population is increasing, we are increasing the production of our food material, but the consumer should accept that food. So related to that this point, I have pasted one of the bloom grape resolving myth, the screenshot of the news which I collected from that it has been published by the director of the ICAR, and they have cleared about the myth of the what is the myth of the bloom grape resolving, that everyone should has known about this type of white layering is nowadays is coming on the grapefruit. So it is what is going on and the myth is going on in the market, that it is the layer of the pesticide or insecticide. So we should not eat the food the grapefruit directly without washing it. But the scientist has proved that this is not a layer of insecticide or a pesticide. It is a layer of a wax, edible wax, which is pasted or which is applied on the fruit. It's a grapes fruit to improvise its quality and to make it insect resistant or pest resistant. So here that is that the wax is known as the bloom. The white layer is known as the bloom. So there is the myth around it. So this type of myth is there with the is associated with so many of the vegetable crops, whereas the cereals and the fruits. But we should not consider this myth. We should identify the what is the actual condition is there and behind actual condition is there behind that probable whether it is a pesticide or insecticide layer or whether it is the wax edible wax has been applied on that to improvise the quality. So the lack of effective leadership, poor funding of the agricultural biotechnology research and development. The biggest problem I must say here that the poor funding of the agricultural biotechnology research. So many colleges and institutes are running their biotechnology and microbiology background, but they are not capable enough. And the funding agencies and the funding criteria, so many funding, I must say that so many funding agencies are present in India and globally, but awareness is not there. The students are not aware that through which direction we should go, and the here the many developing countries overweigh the concerns that the technology might pose a danger to the biodiversity, health, and local jobs. Because the biotechnology is the field where the students learn about each and everything. If we talk about the botany part, that the zoology part, and then coming to the advanced part of the plant biotechnology, animal biotechnology, stem cell biotechnology, IPR and ethics, then the entrepreneurship. But the basic science students should not learn and do not learn these things, this type of ultimately. But what the local industries, local health peoples, and what they do, they require the ultimately core botany persons, core zoology persons. So this is the ultimately a drawback of with our field that I must say that I am also facing this type of uh, problems in my career also, that everyone wants the core botany, but a biotechnology students has more knowledge than the botany and zoology persons, and they can bring a good research to the environment they can produce. But there is a lack of awareness in the community, then the lack of knowledge about the funding agencies, about the colleges, about the societies like Microbiological Society of India. They provide a good platform to us to provide give lectures on that, and so many things are going on in through this society, but lack of awareness is there. Due to lack of awareness, the students are unable to join. Then the agri-informatics coming to overcome these problems, there is the another terminology has been introduced, that is the agri-informatics. Has here I have one challenge has been, that is the lack of research focus and infrastructure. Whenever we go in the laboratory experiment part, it requires lots of manual work, lots of chemicals consumption, lots of electricity, and each and everything is should be consumed. But in case of the agri-informatics, this cost of the experiment is reduced by the half, or you can say the nil, because you just require a good background of your computer knowledge, your then you require a good nowadays the internet is going on the boom, it's boom. So in a good connection of the internet and the good knowledge of the databases only. So informatics is the agricultural biotechnology and the information technology. Here, so many farmers, and you have come across so many tools and techniques, or the many soil. Where here is the health card issue होता है गवर्नमेंट ऑफ इंडिया की तरफ से each and every farmer को. They need to do what they need to do. They need to give the specific data about their soil and click the photographs of that soil. It will generate a report, or it will generate a health card. In that they can gather all the information there, which type of soil is present you have in your field, how much which type of crop you should take to get a good output from that your agricultural expertise. Okay, so it is not like that कि आपके पास में अगर कोई soil मतलब clay जिसमें content ज्यादा है and you are taking the another crop, and you are drought resistant, you are taking the drought resistant crop in that. And if your loamy soil is there and you are taking the you are cultivating those plants which requires the more water. So this type of health card has been issued by the government of India, and every state there are the so many other applications are also present that they gives the virtual assistant to farmers, like the for an instant Huzocan and Osanen 2018 demonstrated how wearable augmented reality technology could guide users to soil sample locations based on the soil map created from the drone imaging. Using the drones, they are plotting the graphs and analyzing the map. They are generating the soil maps and advising the farmers to which type of crop will be usable to your for your crop. Here the terminology comes under the that is the big data. What is the big data? Data is the actual the genomics. It comes under the category of the genomics, transcriptomics, metabolomics. That is each related to the genes, proteins, its pathways, the pathways activation, each and everything. It comes under the big data demand. And the nowadays the demand of this field is increasing because nobody wants to do to waste its money. The bioinformatics tools and the bioinformatics, AI and ML. AI is the artificial intelligence and the machine learning. Then how on this basis the ML is the part of machine learning is the part of the artificial intelligence, and artificial intelligence is the technique, you can say the robotics. Many of the countries has introduced the robotics in their field. They are drone imaging, multiple, I must say here, I have attended one conference in Mau, which has been conducted by the ICAR. There, in Uttar Pradesh, they have presented where the scientist has presented the application of the drone, how they put the for the synchronized farming and the pesticide behavior. They are presenting and applying the drone in their field. So it reduces the manpower, electricity consumptions, and many more. And the data here, if we talk about the machine learning, the how and which algorithm it works, it collects the data, whatever the data we gathered from our literature review, we provide and to the algorithm and generate a model. Whatever the model, when we यहाँ पे कुछ टाइम देना पड़ता है उसको मॉडल को डेवलप होने के लिए, और उसका इंटरेस्ट मतलब उसको वार्म अप कंडीशन होती है जनरली यहाँ उस जगह पे. एंड it will on that basis whatever the data we are producing and whatever the algorithm we are applying, it will generate an output. On that basis it will predict the output, and we can must say try to understand here one technique, that if we want to test the an antibiotic against suppose not a single antibiotic, we need to test the 10 antibiotics, suppose, against an organism. Then what we need to do is test antibiotic assay test for each and every antibiotic against the microorganisms. If we provide the data from the literature review that this antibiotic will work in this concentration and this antibiotic will work on the 25 micro mg per ml concentration, and like this, we provide the specific temperature range, we provide the specific pH range, and we all these things we applied in the model. Then on that basis, whatever the data we are providing, on that basis it will predict the output. It will select one or two antibiotic from that data and say go with this go with this parameter. These are the parameters that specific antibiotic use this concentration, specific concentration, use optimum this optimum pH will be 7.5, and then the temperature will be 25 to 27 degrees centigrade, and many more things it will suggest us. So it will reduces our laboratory experiments time, what we are consuming and we are providing, and in the laboratory experiments it will reduce that. Then the significance of the agri-informatics, as I said, that it is directly related to the artificial intelligence, robotics, and machine learning, and the computational tools. Here are the precision farming and the smart agricultural practice has been introduced. Many of the farmers are using that smart, with the help of the smartphones, they are downloading so many, installing so many agricultural farming related tips and techniques, and they are applying in that their field. Enhancing crop breeding and biotechnology, AI and ML in the pest and disease management. This AI and ML, since last 5 years, there are so many research papers has been published on this technique. Even in the on the antimicrobial drug resistance, also there are the so many papers are available. The scientists have developed a model, mathematical model, to calculate the how the specific antibiotic can go and treat attack on the an specific type of pathogen. Okay. So you can say here it is the advanced version of bioinformatics, not the biotechnology only. It is the advanced version of bioinformatics. The climate change adaptations and risk management, it is a basic most prominent significant of significance of the agri-informatics, where the students uh, here the where the farmers nowadays the climatic conditions are they do not rely on the basic climatic conditions. Through the help of the agri-informatics, they should they collect the data. They put the what here in my area, ये यहाँ का टेम्परेचर असेसमेंट है लास्ट मंथ का, यहाँ पे इतना बारिश हुआ है, यहाँ पे रेनी सीजन है, यहाँ पे हॉट सीजन है। ऐसा सारा पैरामीटर्स जब अपन एप्लीकेशन में अपलोड करेंगे, देन वी गेट द इंफॉर्मेशन that on the climate conditions will change after 3 months, or after 3 hours, you can say 3 hours you may get a heavy rainfall. So on that basis, farmers are taking the cropping and producing the yield of that field. Then they are not taking the or they are doing the synchronized farming or multiple farming also they are doing to make the nutrients available enriched, to make the soil enriched, nutrients rich. Then sustainable resource management, economic and social benefits, it is there. Ultimately what the aim is, our aim is we need to independent enough about the economic independence is should be there, whatever the working we are doing there. This some are the more recent benefits of the agri-informatics, or you can say the agricultural biotechnology. First is the reduces the dependence of the agricultural weather conditions, reduces the cost of materials and resources, then effective maintenance of the health. As I said, the soil card has been introduced, then the increased crop productivity and production. So after this discussion, one question should arise in your mind, that how we can collect this type of data, from what are the sources of to collect that data. So to collect that data about the plant genome, we need to understand about the plant genome. Specifically, if we talk about the bioinformatics part, okay. So we need to. I have introduced in my previous slide that the three terminologies. That first one is the genomics, proteomics, and the transcriptomics. So we need to know that is known as the multiomics, comes under the multi-omics terminology. So this multi-omics, everyone should know about the there is this sequences, then what are the protein structure is formed, then how it is going to be expressed, then in the last part how it is going to be expressed. It should know about the metabolic pathway of that particular gene. So to collect or to gather that information, there are the some databases which are present globally. These are these are known as the related to the plant secondary metabolites, plant genomes, and plant metabolites which are produced by the bacterial or microorganisms. Okay. Out of that, the one you have earlier heard about the PubChem NCBI. So this is the core heart of that database, which provides the each and every information about your genomes, transcriptome, and metabolic pathways. So here are the, I have enlisted few of the databases, that the ensemble plants, phytosome. Some of the plant are, some of the databases are there, like the 3TC tool box, which provides the genomics and breeding databases only for the wheat, barley, and related species, which comes under the 3TC family. And the one genome is one database is there, that is known as the maize GDB, which comes under the genetics, genomics, and the breeding data. Rice genome is there. Then one database is there which provides the about the aspergillus information only. So these are the list of plant genome databases which are related to the plant secondary metabolites, plant genomes, and the plant secondary metabolites. That first one is the most important parameter, and like I love this database personally, that is the Kyoto Encyclopedia of Genes and Genomes, which provides the each and every information from this 2D structure, smile effects, the chemical formula of the metabolite, and the last one is sequencing, and each and everything, whether it is related to the whatever the metabolite is there, whether it is a phenolic, an alkaloid, or whatever, whichever category it comes under, it provides the information to us for that whether it, or it also provides the information about its applications, whether it is a consumable, it is not consumable, it is anti-diabetic potential, it is anti-cancerous and potential. Same information we can gather from the PubChem also. Then CHEY is the bioactive plant compounds and metabolites. One interesting database is the coconut, which provides the information about the natural products, including plant secondary metabolites. Whatever the natural products, because when we work in the case of the plant genomes, there are the plant genome is very complicated, and it produces every gene is producing the multiple proteins, and it is involved in the multiple pathways. So we need to understand the and whatever the plant product is produced at the final stage, they have the complex structure. So the coconut provide the database and each and every information about the natural products. Then the some of the databases which are related to the bacterial secondary metabolites. And here, everyone should know that I have mentioned the bacterial secondary metabolites, which need to know about these things, because they will work in two ways. First, the microorganisms, as you all know, that microorganisms are of two types. One is the good microorganisms, and one is the bad microorganisms. The good microorganisms will be helpful in improvising the crop yield, but the bad microorganisms will attack, will be treated as the pathogen, which is going to be attack on our crop. Okay, so to understand what the mechanisms, whether the microorganism is going to be help us out in the crop productivity, or it is is it going to reduce the crop yield. So the crop, it should be a both. So we need to understand about the genomics of that bacteria. To understand that genomics, that the everyone should know, the bacteria produces a specific type of metabolites which are responsible for the to establish a plant pathogen interaction. Okay, so that to understand that metabolites and the plant pathogen interaction, that the anti-smash database, Dough Biscuit database, Prism, NR Pro, ClusterMine 360, this type of the databases are available. Through that, we can predict or analyze the plant secondary metabolite pathways. Even from the CAG, we can get the information of the pathways. So related to the coming to the my research work related to the agri-informatics, the first one is the identification and management of the etiological agent of Thalia disease of pomegranate. This was my work related to my PhD, and I have completed my PhD on this topic only. In that, I have identified Thalia is the one of the commonly known as the one of the disease of pomegranate, which reduces the crop yield by 60 to 70%, or I must say sometimes if it is in the severe or the last stage of the disease, if the field is get affected by this disease, there is the loss of 100% of your yield. So I have identified the causal organism of that particular disease, and the management through the in vitro level, at in vivo level, and as well as the at field level. Also, I have tried the whatever the formulations I have developed. So in that, during my research part, I got few bacterial strains. This all comes under the category of the Bacillus species, and I identified that the causal organism of this disease is the Collectotrichum species. So that Collectotrichum species has been tested against the bacterial isolates. In the result part, I got this type of plate method has been introduced and developed, that isolates antagonistic activity has been performed. In control, you can see the complete fungus has been grown in the complete plate. But when I have introduced and when I have applied the bacterial strains there is the reduction of the pathogen itself. There is the no further, there is the, so it proves that the some of the metabolites has been identified by the or is produced by the bacteria, or you can say the bacteria is producing few metabolites which are inhibiting the growth of the your pathogen. Okay, so the next time is to next task is to identify this metabolites. Okay, whatever the organic compounds is present and whatever the metabolites are present in that particular bacterial species, to and to purify it and test against the pathogen. It requires the entire efficiency in the downstream processing. Okay, after whether instead of doing that, what I have done, I have collected the data from the review of literature and from the databases, and found what are the organic compounds, what are the metabolites which are present in the Bacillus species and which are responsible for the antifungal activity, not for the Collectotrichum itself, but Bacillus has the antifungal activity against the other species. So I have collected the data and I found that the iturine A and the 2-methyl butonic acid, these are the two main important compounds which are present in an abundant form in Bacillus species and act as a antifungal compound. They have antifungal activity against. So what I have did instead of doing isolating this iturine A and 2-methyl butonic acid from the species and testing against the fungal species at the in vitro level, I have used the informatics here. Information technology here. I downloaded the structure of CFEM protein, that is the specific protein which is responsible for the pathogen, which is responsible for the host pathogen interaction. So I tested against this iturine and 2-methyl butonic acid, the CFEM protein, against and docked it with this protein. So I got the information here that and two type of different protein. First is the apocutinase and the E600 cutinase complex from the CFEM. This were the proteins from the Calectotrichum species, and vanomyl, carbendazim and mancozim mix were the control that as the antifungal compound, or the you can say the pesticides which are commercially available in the market and which are used by the farmers to recover their pathogens or to recover their yield for the management part. Then iturine A and the 2-methyl butonic acid, this chemical structure I got from the PubChem itself. So I docked a molecular docking analysis has been done, and I got the result that the RMSD values, that the Ramachandran plot values and the activity of the manomyl and carbendazim is or iturine A is similar to that. And then the whatever the ligand binding protein you can see here, it is the iturine A is binding at the same place, the manomyl and the carbendazim, their molecular bonding is there. In another section of my work, I have developed as I used one terminology, that is the sustainable farming. So I have substantially sustainable produced one byproduct from the pomegranate fruit, that is the wine, pomegranate wine. And the antidiabetic potential of that byproduct has been developed. What has happened when this disease comes to the field? Sometimes it happens that it occurs before just 2 or 3 days of the harvesting, and and leaves a black spot on the pomegranate peel. So it reduces the market value. When here the farmers when is expecting the 100 rupees per kg in wholesale rate, okay, of the farm it reduces, and has to sell its fruit by 50 rupees per kg or 60 rupees per kg. So to overcome this 40 decline, और यहाँ पे जो 40 rupees का जो loss है उसको overcome करने के लिए I have developed one technique, that is the antidiabetic potential byproduct development using the anthracnose infected pomegranate fruit. So in this, wine has been developed, and at last moment, the LCMS analyzes whatever the essential compounds are present to generate, to find out what are the specific amounts which are specific metabolites which are present in that after the last byproduct has been done, and estimation of the antidiabetic potential has been done. From the LCMS analysis, I got 8 different products or metabolites which are responsible for the antidiabetic potential. These are the name of these compounds, and their compound ID is I got from the PubChem itself. So using that compound, I have stopped my work here only. I just find out whatever I have analyzed over the quality parameters, quantitative parameters, and estimated the antidiabetic potential using by giving the, since it was a fruit juice, so I have directly tested against the patients, diabetic patients. But one of my research scholar or one of my trainee has further carried out this research, and what she did, she selected these compounds, and tested against the enzyme or the gene responsible for the this for the diabetes. Okay, alpha amylase is the protein which is responsible for the diabetes. So it tested against the and the at in silico level. So it tested in the all the 8 compounds has been tested. I have tested the 2 or 3 result of the 2 or 3 here. Then there how this binding is going on, at what chain site they are, what are the ligand binding positions are there of that particular molecule. And in control, aracapose is used. In control, aracapose is the medicine which is generally consumed by the diabetic patients. So here we can see the affinity of the acrabose is the minus 7.531, and the same affinity is given by the CAY10506 compound, that is minus 7.799. So total energy is higher in that case. In that case, the total energy is very, very low, but electrical conductivity, van der Waals energy and electrical energy is very much similar to in both the conditions, and they are residing at the similar kind of many of the common compounds are there, and binding of the at chain A only. So in that way, we can identify, okay, out of that 8, this is the compound, a specific compound which need to be further tested and which needs to be further give in the purified form to the diabetic patients. In another research of one of my research, that is the differential expression of the Indian spices against human pathogens and in vitro level and in silico study. I'm not showing here the results of the in vitro level, showing the results of the in silico level. Here is the different Indian spices. The chemical formulation has been developed of the Indian spices and tested against the your this AMR, antimicrobial drug resistant category, which comes under the antimicrobial drug resistant category, that is the E. coli, S. aureus, staphylococcus, and Klebsiella pneumoniae. They have been tested against that at in vitro level. Then in silico level, what I have done, then molecular docking of the active compound which are present in the Indian spices like the eugenol and cinnamic acid and thymol, and tested against the crystal structure of the antimicrobial drug resistant bacteria like the E. coli and S. aureus of protein. Here a similar kind of values I have got. The score and the total energy was comparable to the control one, that whether this medicine has been introduced to that is has the highest energy. And here we have the total energy 2 and 2b is showing the minus 18.5. This is the activity of the total energy of the eugenol and the cinnamic acid. Then in another study of the molecular docking, we have performed about the antidepressant compounds in the postpartum diet of Rajasthan. You have come across with the case of the one of the so many ladies which comes and which are suffering from the postpartum depression, that is known as the PPD. After the postpartum, after the delivery, due to the sudden decrease in the estrogen level, women goes under the depression category, and sometimes it is इतना 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 सीवियर होता है कि they cause suicide, they commit suicide, they harm themselves, they harm their child, they harm their family. Okay, so to come across, I have established one of my student have did the research on that, that in the Rajasthan, generally after the delivery, a specific type of food is given to the female, that is known as which is the combination of the soa, haldi, salt, satavari, and ajwain. This type of combination of the food is given to the all type of female. I think in all India level, at it all many of the states they consume this type of food. So we've identified and we got the collected the data from the databases and compounds which are what are the different compounds which are responsible for the antidepressant activity present in that specific plants. So came across with the curcumin, gingerol, then the impramine. These are the compounds which are responsible for the antidepressant activity. Further molecular docking analysis has been done and found that as compared to the that juranolone and the sertraline is the two medicines which are given to cover to cure this depression, and these are the all natural compounds which are present in the food, our food, which treats the depression, and only the one single gene is responsible 5-HTT, which is responsible for the cause of this depression. So this all compounds has been treated or has been tested against this particular gene along with the this control compound and got a specific that the test resmosmosol, that the imperine and the strataline that has the minus 9.4 affinity, and total energy and electrical energy is the similar of that. So future scope and conclusion of that, it agri-informatics, bioinformatics set to revolutionize sustainable agriculture and food security. It will provide a good data, good genomics, just requires a good you depth knowledge of that analysis, data analysis, that review of by doing the thoroughly review of literature and doing using the AI tools, we can go with that. Then awareness program and campaigning program should be done to make the students aware about this field and they make their future in this field. And policy makers should give the information about the researchers should present their data nicely, and policymakers should enhance or make the community together, where a platform should be provided by them to that the farmers are are giving the problems to sharing their problems, and the researchers are giving solutions to them. And then investment in the agri-bioinformatics will be key of tackling global agricultural challenges. So I thanks here. I end up with my presentation. Thanks for patient listening. I have thanks in my professor guide to Professor Kanika Sharma and Dr. Anash Marvel at Department of Biotechnology, MLSU. I am extremely thankful to Dr. Arvind Deshmukh and Mr. Sarath for providing an entire organizing team for conducting such an international good webinar and providing me the platform to present my research work. Then my director of Faculty of Science, Dr. Sapna Srimali, for allowing for allowing me to present my research at such international level. Then my trainees, Ms. Dona, Ms. Bhavya, and Ms. Aarti, and last my parents and my in-laws. Thanks to all of you.
Thank you. Thank you so much, ma'am. Do we have any questions? As I can see, one question is there. Is there any scholarship for international students in your university?
No, in our university, we are not providing any scholarship, but we can plan, we can arrange a training program for them.
Okay.
Uh, so my email ID is given on this page, and whatever the
Yes.
query is there, they can
Yes.
contact me on that.
Yes. Yes. Correct. Okay. So I would request Miss Janvi Goel to please proceed for the vote of thanks. Janvi, are you there? Janvi, are you there? Uh, okay. I have to unmute them now.
Yeah.
I think she is not here. I guess. Okay, no issues.
Sarath, sorry for interruption. Here from this
Yes.
platform, I want to introduce one thing that our university provide the internship program for
Yes.
in the field of the bioinformatics. So any students from the BSc, MSc or PhD scholars are there can contact me on this email address. Or if any student is wants to do research in this field, or if wants to perform make their career in this field, or any other information they want to know about from me, they can directly contact to me from that.
Yes. Thank you, ma'am.
Okay.
Miss
Okay.
Janvi, please switch on your microphone if you are able to hear me. Uh, yes. Kindly try now. Kindly switch on your microphone. Yes. Now you can switch on your camera and proceed for the vote of thanks, Miss Janvi. Miss Janvi, please switch on your camera and proceed for the vote of thanks. Janvi, try now. I have sent you a request to unmute yourself. Janvi, speak something. You are not audible. Hello, Janvi, you are not audible. Hello, you are not audible, Janvi. Okay, no problem. Maybe you are facing some problem, network problem or something like that. Rashi, are you there? I will ask you to unmute. Rashi, are you able to unmute? Speak something. Rashi, hello. खुशबू मैम आपको आवाज आ रही है क्या?
Yes, sir, you are audible.
Yes, sir, I'm able to unmute. Can
Proceed.
everybody hear me?
Yes, proceed.
All right.
Proceed.
Okay, good evening, everyone. So it is my absolute honor and privilege to deliver the official vote of thanks for this wonderful international webinar on agricultural biotechnology. As we come close to an enlightening and thought-provoking gathering, I want to express my deep gratitude to one and all who made this webinar on agricultural biotechnology a remarkable success. First and foremost, my sincere thanks to our keynote speaker, Dr. Khushbu Jain, Assistant Professor, Department of Biotechnology, Faculty of Science, Janardhan Rai Nagar, Rajasthan Vidyapeeth, deemed to be University, Udaipur, Rajasthan, who gave a very informative lecture on the topic of agricultural biotechnology. Then I extend my heartfelt thanks to the organizing board members. Firstly, Professor Dr. Arvind M. Deshmukh, Professor Emeritus and the former head Department of Microbiology, Dr. Baba Saheb Ambedkar, Marathwada University, Maharashtra, Government of Maharashtra, the founder and All India Chairman of Microbiologists Society India, and secondly, Mr. Saratsuva Paine, the national representative of Microbiology Society, who had highlighted the direction for organizing this wonderful program. Lastly, I truly thank our dearest participants who spent some time attending our international webinar today. We are very grateful to all of you. Thank you all for joining us, and I hope to see you in upcoming sessions. Thank you so much.
Thank you, Rashi. Thank you,
Yes,
Sarat.
thank you everyone. Thank
Yeah.
you.
Yeah.
Thank you.
I'm leaving now. Okay, bye.